Astrocytes are the most abundant glial cell type in the central nervous system, and they perform a remarkable breadth of functions. Their cell bodies emit numerous processes that radiate outward to surround blood vessels, envelop synapses, and fill the spaces between neurons throughout the brain and spinal cord. Two morphological subtypes exist. Protoplasmic astrocytes are found in the gray matter and have thick, extensively branched processes. Fibrous astrocytes are found in the white matter and have longer, thinner processes. A defining structural feature shared by both subtypes, and one that distinguishes astrocytes from all other glial cells, including microglia and oligodendrocytes, is the presence of vascular end feet. These are specialized tips of astrocytic processes that physically wrap around the walls of blood capillaries, placing astrocytes at the blood-brain interface where they contribute to barrier function. Astrocytes express both voltage-gated and ligand-gated ion channels, and through these they actively regulate extracellular potassium and calcium concentrations in the interstitial space. Since fluctuations in these ion levels directly affect neuronal excitability and firing, astrocytes effectively modulate neurotransmission through this buffering role. They also support neurons structurally and metabolically by producing extracellular matrix components such as laminin and by releasing growth factors including nerve growth factor, fibroblast growth factor, and brain-derived neurotrophic factor. At sites of injury, astrocytic end feet at blood vessels release cytokines and chemoattractants to coordinate the immune response to CNS damage. When the brain is injured, astrocytes undergo a characteristic transformation called reactive astrocytosis. They increase in size and in some cases multiply in number. This response involves upregulated expression of a structural protein called glial fibrillary acidic protein, GFAP, and the synthesis and release of inflammatory cytokines. These cytokines orchestrate local inflammatory responses and attract immune cells from the blood into the CNS. Astrocytes also serve a critical neuroprotective function through glutamate regulation. Glutamate is the brain's most abundant excitatory neurotransmitter, and at high concentrations it becomes toxic to neurons, a phenomenon called excitotoxicity. Astrocytes defend against this in two complementary ways. 1. Glutamate transporter proteins sitting in the astrocyte cell membrane actively capture glutamate from the synaptic space, removing it before it can accumulate to toxic levels. 2. Astrocytes contain an enzyme called glutamine synthasase, which converts the collected glutamate into glutamine, an amino acid. This enzymatic step effectively detoxifies the CNS of both glutamate and ammonia simultaneously. It is important to know that neurons grown in cell cultures die when exposed to high glutamate concentrations, unless astrocytes are present.